Yes, yes, we are live now. Welcome to Zoology Online Guru, my YouTube channel. Since 3 days, we are discussing about human digestive system, and today our topic is pancreas and physiology of digestion. Okay, come and join the live session. We will start exactly at 9 o'clock. Come and join the live session. Up to yesterday, what we discussed, once again recall, the human digestive system is divided into two parts. One is alimentary canal and associated glands. This division is for our convenience to remember perfectly, to not to confuse. We divided it into two parts. One is alimentary canal, second one digested glands. Alimentary canal starts with the mouth and ends with anus. The digestion starts in the mouth and completes in the small intestine ileum. The alimentary canal parts are mouth, buccal cavity, pharynx, esophagus, stomach, small intestine and large intestine. And the associated glands, the glands extra alimentary canal and within the alimentary canal. The extra alimentary canal glands are liver, pancreas and in the buccal cavity salivary glands. If you are able to listen my voice, okay, if you have any disturbance in the clarity of the voice and clarity on the board, let me know immediately. Tell me, raise your hand, comment, chat with me if there is any problem. Coming to the digestive glands, digestive glands are they are within the alimentary canal, gastric glands and intestinal glands. Gastric glands present in the stomach, intestinal glands present in the intestine. And coming to the extra alimentary canal, digestive glands are in the buccal cavity salivary glands. In the humans, we have 3 pairs of salivary glands. Salivary glands secretes saliva. Another largest gland in our body is liver. Yesterday we discussed about the liver and its functions. Now, today our topic is pancreas. After discussing the pancreas, then we go to another topic, that is physiology of digestion. If you have any doubts, ask me, raise your hand in the chat box. We will discuss that doubts also. Okay, come on, join my YouTube channel, Zoology Online Guru. We also have our own app, Kumar Institute of Zoology, and my Telegram channel name is Kumar Institute of Zoology YouTube channel. There you can find number of PDFs are there. Whatever we are discussing, all those files in the form of PDFs, they are available there. Go through those PDFs. Otherwise, you can download those PDFs. And our own app is Kumar Institute of Zoology. Okay, and my YouTube channel name, Zoology Online Guru, the one and only zoology related YouTube channel. If you have any doubts in the zoology, you can ask here, raise your hand. Mostly we will discuss in the live channel, live session. Every day we have a live session, 9 p.m. to 10 p.m., 1 hour live session. Today our class is about the pancreas and physiology of digestion. Okay, now we'll start our topic with pancreas. Pancreas, yesterday we discussed about liver. Okay, our topic is human digestive system. So in the human digestive system, now our topic is pancreas. Pancreas. Pancreas is the second largest gland. Pancreas is the second largest gland in our body. Second largest gland. First gland is liver. Second is the pancreas. Pancreas is the second largest gland. Location. Where pancreas located? Location. In the curvature of duodenum. In the curvature of duodenum. They secretes pancreatic juice. Pancreatic juice. Okay. And see, come to location here. So this is duodenum. Duodenum is U-shaped in structure. It is U-shaped. In the U-shaped structure here, the location of pancreas in the curvature, it is located here. It is a pancreas is a leaf-like gland. So shape. If you take shape here, leaf-like gland, second largest gland. Special category. The specialty of pancreas is it is a mixed gland. It is a mixed gland. What is the meaning of mixed gland? Mixed gland means pancreas in its cross section, it shows two types of cells. It shows two types of cells are present in the pancreas. One is pancreatic acini cells. Pancreatic acini or acini cells. Second one, pancreatic Langerhans cells. Langerhans cells. So these two types of cells are present, and the two behave differently. So it is the mixed gland. Mixed gland means here they can secrete enzymes and also hormones. If a gland secretes enzymes only, that gland is called exocrine gland. Exocrine gland. So in our body, glands are two types. Glands are two types. Glands are, there are two types. One is exocrine glands. Second one, endocrine glands. Exocrine glands, they are also called duct glands. Duct glands, they have a tube or a duct, and through that duct, the liquid, the juice, they enters into the target organ. So here ducts are present, otherwise tubes also present in the exocrine glands. Okay, after releasing of their secretions, the secretions enters into the tube. Along the tube, they reach the target organ and their functions there. And what they release? They release mostly enzymes. Here in the point of pancreas, they release enzymes. So here exocrine gland or exocrine system in the pancreas. Okay, and here in our body there are two types of glands are there. One type is called exocrine gland, second is called endocrine gland. Exocrine gland, also called duct glands, they have tubes. From the tubes, enzymes are released and enters into the target organ, and the enzymes participate in the digestion of the food material, digestion of food materials. Okay. Second one is this is endocrine. Endocrine means they are ductless glands. Ductless glands means the tubes are absent. That is why they released directly into the blood. So ducts are absent, tubes are absent, and the target organ is very far away in the in our body by location. So to reach that target organ, they directly enters into the bloodstream, and through the blood they are transferred to the target organ, and at the reaching of the target organ, they enters into the target organ and activates that gland to produce or release that certain enzymes or hormones. So here in the case of endocrine glands, ductless glands, they release specially hormones. So they release hormones. Enzymes and hormones both are proteins. Okay. Don't forget, both are proteins. Enzymes also proteins and hormones also proteins. Here, endocrine glands, ductless glands, they produce hormones. They directly released into the blood. Through blood, they are transported to their target organ, and they activate that target organ. That is hormonal action. So here, our pancreas has these two characters. So pancreas produces. You see the pancreas, okay? Pancreas produce enzymes and also hormones. Pancreas produce enzymes and also okay, Madhukar Reddy. Yes, okay. Welcome to Zoology Online Guru, all of you, and share this live session link to your friends and those who deserve to learn zoology. Okay, welcome to Zoology Online Guru, my YouTube channel. So we are discussing about pancreas. Pancreas is now, it is also known as mixed gland. Why we are calling this pancreas as a mixed gland? Because it secretes enzymes and also hormones. And the glands which secretes enzymes, those are called exocrine gland. And the glands which secretes hormones are called endocrine glands. But pancreas secreting enzymes and also hormones both. That is why it is known as mixed gland. One and only gland in our body is the mixed gland is pancreas, only mixed gland. Okay. And coming to the microscopic structure, if you observe under microscope the cross section of pancreas, we observe different types of cells. See, some cells are unicellular and they are looks like this. So like that, there are so many unicellular cells are there here inside. So they are these cells are known as acini cells. They are known as acini cells. Acini cells. And some cells discovered by Langerhans, and those cells are so these type of cells also present there in the pancreas. So here these cells are known as Langerhans cells. Langerhans cells. Langerhans cells are of two types, alpha cells and beta cells. Alpha cells and beta cells. Beta cells, they produce insulin hormone, insulin. And alpha cells, they release glucagon. These two are hormones. So insulin and glucagon, these two are hormones. And acini cells, these acini cells, they release enzymes. they release certain enzymes. They enter into the duodenum and they digest different types of carbohydrates, proteins, and also lipids. So here SNI cells and Langerhans cells, both type of cells are present, and SNI cells are secreting enzymes and Langerhans cells are secreting hormones. That is why pancreas is known as mixed gland. It secretes enzymes and also hormones. But our topic in this lesson, our topic is digestion. So now we are discussing about only SNI cells and the enzymes present in the pancreatic juice. So different types of enzymes are present in the pancreatic juice. Okay, the enzymes present in the pancreatic juice are trypsinogen, chymotrypsinogen, okay, procarboxy peptidase. So like that, and as pancreatic lipase, okay. So like that, here protein digesting enzymes, carbohydrate digesting enzymes, and pancreatic amylase. See here, pancreatic amylase. We already discussed it in the buccal cavity. Salivary glands, they secrete saliva. Saliva also contains an amylase that is called ptyalin, also known as salivary amylase. Salivary amylase converts carbohydrates into maltose. So here pancreatic amylase also converts the carbohydrates, that means polysaccharides, into disaccharides. So here pancreatic amylase, lipase, carboxypeptidase, chymotrypsinogen, and trypsinogen. So all these are the enzymes present in the pancreatic juice. So here up to now we discussed alimentary canal and digestive glands. Up to now, any doubts? If you have any doubts, ask me, raise your hand, chat with me. We will take 2 minutes of time for the doubt session, and then we enters into another new topic, that is the physiology of digestion. Up to now, glands and alimentary canal were completed. Any doubts? Ask me. One second. Yes, okay. Now our topic is now we enter into the physiology of digestion. The physiology of digestion. First, in the alimentary canal, buccal cavity with the teeth and salivary glands, when we take food material into the mouth, we masticate the food materials by using different types of teeth, that is incisors, canines, premolars, and molars. We already discussed about these different types of teeth. And after mastication, the food is mixed with saliva. Saliva contains ptyalin enzyme, also known as salivary amylase. And salivary amylase with carbohydrates, they convert carbohydrates into their simplest form, maltose. Here, what is the meaning of digestion? First, if we see what is the meaning of digestion here, conversion of complex non-diffusible conversion of complex non-diffusible food substances into simple diffusible or absorbable. Okay, complex non-absorbable food substances into simple absorbable food substances. That is known as digestion. So digestion starts in mouth. So mouth is the first part, that is buccal cavity. In the buccal cavity, food is masticated. Okay, in the food we have three different types of food materials, that is carbohydrates. Whatever we eat every day, that food material contains the macromolecules, carbohydrates. Macronutrients, carbohydrates. Food material are two types, micronutrients and macronutrients. We are discussing about the macronutrients because digestion means complex food materials we have to discuss. So here carbohydrates, proteins and fats are present, or lipids. Lipids are present in the food materials. So conversion of these complex food materials into their simplest form. Carbohydrates simplest form is glucose, fructose, galactose. So carbohydrates complex food molecules converted into their simplest forms, glucose, fructose and galactose. Proteins are complex, and proteins are made up of amino acids, and fats. Fats also the largest complex food molecules, macronutrients, and they are converted into glycerol and fatty acids. So this is digestion. So carbohydrate digestion produces glucose, fructose, galactose. Protein digestion produces amino acids, and fat or lipid digestion produces glycerol and fatty acids, and all these materials are known as digested food material. Digested food. And this digested food is present in the small intestine. The part is called ileum. So in the small intestine, ileum is the part where digestion completes. Digestion starts in the mouth, buccal cavity, and completes in the small intestine, ileum. Coming to the first one, the digestion in the buccal cavity. In the buccal cavity, teeth and salivary glands are present. Salivary glands secretes saliva. Saliva contains ptyalin enzyme. That enzyme converts carbohydrates into maltose. Very simple. First one, we are discussing about the digestion in the buccal cavity. So first here, digestion in the buccal cavity. Digestion in the buccal cavity. Digestion in the buccal cavity. This is digestion. Digestion in the buccal cavity. In the buccal cavity, teeth are present. Teeth and saliva. The pH of saliva. Here pH most important, and certain enzymes, they work at a certain pH only. So at every point or every stage we have to know the pH of that juice and then the activation of the enzymes. So certain enzymes, they work only at a certain pH. Here the saliva pH is 6.8. 6.8 is the pH of saliva, and in the saliva salivary amylase is present. Salivary amylase converts carbohydrates, so here ptyalin also known as salivary amylase. So salivary amylase, ptyalin converts into maltose. Here carbohydrates are polysaccharides. So polysaccharides are converted into their disaccharides. So next, in the next step, disaccharides converted into their monosaccharides like glucose, fructose and galactose. But in the mouth, buccal cavity, this is the reaction takes place. Carbohydrates, that is called polysaccharides, converted into maltose. But the bolus, bolus is the food material. Bolus is the masticated food material. Masticated food material mixed with saliva, and ready to swallow. So after mixing with saliva, mastication, okay, breaking, a churning movement of the teeth, so all make the round shaped ball like structure of that food material in the mouth, and that round shaped food material ready to swallow means that present at the end of the buccal cavity, and by using the tongue it is pushed backside into the pharynx area. So up to there, the round shaped structure of the food material that is present in the buccal cavity, that food material is called bolus. So bolus, in the bolus, what happening here? Only carbohydrates. Actually the food material contains 3 macronutrients: carbohydrates, proteins and fats. But in the buccal cavity, only carbohydrates are partially digested. Partially means polysaccharides converted into disaccharides, not the monosaccharides. So digestion starts in the mouth or buccal cavity, and in the buccal cavity carbohydrates only partially digested and no change of proteins and lipids. So up to here, this is known as digestion in the buccal cavity. Up to now, any doubts? Ask me. Up to now, any doubts? Okay. Right. Now, next is the digestion in the stomach. We'll discuss. Next topic is second one. Digestion in the stomach. Digestion in the stomach. We discussed already in the elementary canal topic. Stomach is with 3 parts: cardiac stomach, fundic stomach and pyloric stomach. Cardiac, fundic and pyloric stomach, and they secretes certain mucus and proenzymes and HCl and Cassell's intrinsic factor. So all they collectively called gastric juice. So collectively called gastric juice. So the enzymes present in the gastric juice, okay, and the pH is most important here. pH of the gastric juice because of the HCl very strongest 0.9 to 1.8. See, very much strongest acid. Okay, 0.9 to 1.8 is the pH of a gastric juice. Okay, here mucus lining is present inside the wall of the stomach, and this mucus protects the effect of HCl on our own stomach. So stomach also made up of muscles, so HCl and enzymes may act on our own stomach. So here the thick mucus layer covers entire inside the stomach wall, and that protects from the highest pH of 0.9 to 1.8. and gastric juice, in the gastric juice here proenzymes are present, proenzymes. I think green color also is visible to you, very nice. Okay, green and yellow. Okay, so proenzymes are present here. The proenzymes are pepsinogen. The first proenzyme is pepsinogen, is a proenzyme. Another one, prorenin. So here whatever the HCl is present in the stomach, this HCl converts the inactive, pepsinogen means here inactive. So wherever you find gen, gen means inactive enzyme, and pro also inactive enzymes. So first these inactive enzymes, first they convert into active form. So here HCl is required, or HCl converts inactive enzymes into active enzymes. So pepsinogen converted into pepsin, and here prorenin in the presence of HCl, okay, and converted into renin. And once pepsin is created by the action of HCl, once pepsin is created, the remaining pepsinogen converted into pepsin. So this is the starting problem. So starts the kick, to start the bike, kick is important. So here HCl kicking the pepsinogen to convert into pepsin. Once pepsin created, the total pepsinogen whatever is present, that is converted into pepsin. So this reaction is called autocatalysis, self-activation. Autocatalysis. This is called autocatalysis. So once pepsin is created, this pepsin converts the remaining pepsinogen into pepsin. So here what happening now? The inactive enzymes are converted into their active form. So inactive enzymes are converted into active form. Now what is the function of a pepsin and renin, we'll discuss. So here, up to here, any doubts? Up to here, any doubts? Up to now, okay. Now we'll discuss about the first enzyme, pepsin. So the proteins present in the food material, in the presence of pepsin enzyme, in the presence of a pepsin enzyme, they convert into proteases and peptones. This is the one reaction. Means proteins are digested in the stomach. Okay. First active enzyme pepsin, it converts the proteins into proteases and peptones. And second reaction here in the stomach by milk protein. The protein present in the milk is casein. The protein present in the milk is protein, and milk protein casein in the presence of renin. And calcium ions. In the presence of calcium ions, they convert into calcium paracasinate. Calcium paracasinate. Calcium paracasinate. Now, this calcium paracasinate, calcium paracasinate, now it converted into peptides or peptones and peptides. Peptones and peptides. See, this is the reaction here. See, first proteins are converted into proteases and peptones. Now the milk protein, that is called casein, converts the milk protein casein into calcium paracasinate, that is also known as curdling of the milk in the presence of calcium plus 2 ions. In the presence of calcium ions, the renin converts the milk protein casein into calcium paracasinate, and now this calcium paracasinate in the presence of pepsin, pepsin, they convert it into peptones and peptides. See here, these peptones, peptides and proteases, peptones, all are called here the digested, protein digested end products. They are called end products. End products of proteins. End products of proteins in the stomach. In the stomach. And now here we used only protein words. So proteins and the milk protein and calcium paracasinate also a protein. So here only in the stomach. Stomach is the place where proteins only digested partially. Proteins only digested partially in the pH of 0.9 to 1.8 is in this pH the pepsin and renin, they converts the proteins into peptides, peptones and proteases. So this means here stomach partially digested the proteins, buccal cavity partially digested the carbohydrates, and coming to the next third macronutrient is lipids. So there is no change of the lipids in the mouth or buccal cavity and in the stomach. Means lipids are digested only in the small intestine. Okay. So now, up to now, any doubts? If you have any doubts, but the digestion in the buccal cavity and digestion in the stomach, up to now, if you have any doubts, raise your hand. Okay. Now, the last part in the physiology of digestion, the last part is called the digestion in the small intestine. Digestion in the small intestine. The digestion in the small intestine. See here, liver with gallbladder, cystic duct, okay. Duodenum, pancreas. So liver, here pancreas. These are the cells present in the small intestine, duodenum. So liver secretes bile juice, pancreas secretes pancreatic juice, and the small intestine secretes intestinal juice. Intestinal juice. So here intestinal juice pH, the pH of the intestinal juice is 7.5 to 8.0, basic in nature. So basic in nature. Startingly in the mouth 6.8. Next stomach acidic in nature 0.9 to 1.8. The small intestine it is the 7.5 to 8.0. So alkaline. So the alkaline in nature. So the pH, okay. So the intestinal juice is alkaline because the pancreatic enzymes all are work or function at alkaline pH 8.0 of pH, the pH 8. They work at the pH 8 only. That is why the intestinal juice, the pH is 8.0 or so 7.5 to 8.0. So here at the case, from the liver, gallbladder enters into the duodenum in this area. From the pancreas, pancreatic juice also enters into the duodenum, and intestine, from the intestine, intestinal juice also enters into this area. Means intestinal juice, pancreatic juice, bile juice. So bile juice, there is no enzymes. Okay, so there is no any matter about the digestion of food materials. So bile juice left it. Okay, and coming to two important juices are there: pancreatic juice and intestinal juice. Coming to the first one, pancreatic juice. So the digestion in the small intestine, we converted or we divided, for our easy to understand, we divided it into three major heads. First one is digestion of digestion of carbohydrates. Digestion of carbohydrates. Digestion of carbohydrates. So here pancreas. So pancreas is with pancreatic amylase. Pancreatic amylase. Pancreatic amylase converts polysaccharide, that's called carbohydrates. Carbohydrates into disaccharides, maltose, sucrose, maltose, you can make a plus here. So here maltose plus sucrose plus lactose. They are the disaccharides, converted into disaccharides. So pancreatic amylase converted carbohydrates, they are called polysaccharides, into their disaccharides. Now each one, once again, they are converted into their monosaccharides by their specific enzymes. Here maltose, second one is here sucrose, lactose. So here these three disaccharides, they are converted into their monosaccharides by maltose, converted into maltose by enzyme maltase, sucrose by sucrase or invertase, lactose by lactase. See the substrate and enzyme. So substrate, basing on the substrate name, the enzyme name we discover here. See, simple. Lactose is substrate, is digested by lactase. Ase is the enzyme. Ase, wherever you find the suffix ase, that is enzyme. Ose, ose means sugar. So here maltose sugar, maltase enzyme converted this into glucose and glucose, two glucose molecules. Two glucose molecules are monosaccharide, collectively called maltose. Here sucrose is the combination of one glucose and one fructose. Glucose, fructose is called sucrose, and lactose is a combination of glucose and galactose. So glucose, galactose are known as lactose, and glucose fructose is known as sucrose, and glucose two glucose molecules are collectively called maltose. See here, so two glucose molecules, one glucose and one fructose, and one glucose and one galactose. So these are the end products of carbohydrate digestion in the small intestine by the intestinal juice, and here pancreatic juice. In the pancreatic juice, here pancreatic amylase is present, and disaccharidases. Disaccharidases is the enzymes present in the small intestine. So here disaccharides are maltose, sucrose, lactose. Maltase, sucrase, lactase are the enzymes here. These enzymes are known as disaccharidases. Disaccharidases. Okay, these disaccharidases enzymes are present in the intestinal juice. So pancreatic juice converts into carbohydrates into maltose, sucrose and lactose, and then intestinal juice they converts into their monosaccharides. Clear now? So this is the first step in the our topic here, digestion in the small intestine. So in the small intestine, total digestion completes. That is why we have to discuss each and every macronutrient. First, carbohydrates, and all the carbohydrates, 100% all the carbohydrates are converted into their respective monosaccharides, glucose and fructose and galactose. So this completes the carbohydrate digestion in the small intestine. Second one, protein digestion. So in the small intestine, second one, digestion of proteins. Proteins already partially digested in the stomach by pepsin. Yes, so by using the pepsin in the stomach, the proteins are partially digested into here proteases, peptones and peptides. So these proteases, peptones, peptides along with some of the undigested proteins. So proteins, proteases, peptones and peptides all are present in the food material, enters into the small intestine, and they mixed with the intestinal juice and pancreatic juice. Okay, in the intestinal juice, the enzymes are tripeptidase, dipeptidase and carboxypeptidase. See here. First, see small intestine, intestinal juice, also known as succus entericus. Succus entericus is the intestinal juice. In this juice, the enzyme activator is present. Like HCl in the stomach, here another enzyme activator present, that is called enterokinase. Enterokinase is the enzyme activator. Here this enterokinase activates the inactive enzymes present in the pancreatic juice. Trypsinogen, gen means inactive, chymotrypsinogen, procarboxypeptidase. So pro means inactive. So all these are converted into active form by the enterokinase. So it is called enzyme activator. And another enzymes present in the intestinal juice. The enzymes present in the intestinal juice are, first enzyme, that is called tripeptidase. Second enzyme is dipeptidase. And third enzyme is aminopeptidase. These are in the small intestine, succus entericus. And whatever the pancreatic enzymes, pancreatic juice enzymes. Pancreatic juice enzymes are first number one, trypsinogen. Second one, chymotrypsinogen. And third one, so in inactive form, pro-carboxypeptidase. So these enzymes are present in the intestine, and whatever the food enters into the intestine with the proteins and proteases, peptones and peptides, all these enzymes collectively convert that proteins, peptones, peptides, proteases into simplest form amino acids. So up to now, any doubts? Up to now. So what are the two different types of enzymes present in the intestine? Intestinal juice and here pancreatic juice. First we discuss about this trypsinogen, chymotrypsinogen, and then it enters into the remaining one. Okay. So next here, first enzyme here, whatever the proteins present in the small intestine is called here trypsinogen, okay, and chymotrypsinogen, and pro-carboxypeptidase. All these three are inactive form. All are inactive forms. Inactive. So they are converted into their active form by enterokinase. So the enzyme activator converts them into trypsin, chymotrypsin, and carboxypeptidase. So all these are active form. Here trypsin, chymotrypsin, carboxypeptidase. First come to these two, trypsin and chymotrypsin. Trypsin and chymotrypsin. So whatever the proteins present in the undigested proteins enters into the small intestine, these undigested proteins are converted into their proteases plus tripeptides, protease and tripeptides. So the proteins are converted into proteases, tripeptides and also peptones. See, so proteases, tripeptide, peptones all are created by the action of trypsin and chymotrypsin. Now coming to here, the carboxypeptidase, okay, and before the small intestine, the succus entericus contains tripeptidase, dipeptidase also. So here aminopeptidase also is there. So remember this one. First tripeptidase. So whatever the tripeptides they are created, so here tripeptides, they are converted into dipeptides and amino acids. So here one amino acid, second amino acid, third one. This is known as tripeptides. These tripeptides, they convert into dipeptide and a single amino acid by the action of the tripeptidase. Tripeptidase present in the succus entericus. Now the second, the dipeptidase. Now the dipeptidase or these dipeptides next converted into amino acids by an enzyme dipeptidase. So dipeptides means double, two amino acids, they convert into single mono amino acids. Here dipeptides are converted into amino acids by dipeptidase enzyme. Okay, next coming to the aminopeptidase and carboxypeptidase. See here, aminopeptidase and carboxypeptidase. These two converts whatever the peptides and peptones and whatever the peptones are present. So here peptide means here group of chain of amino acid. So every amino acid have two ends. One is amino end and second end is called carboxylic end, COOH end. Okay, and here aminopeptidase, they cleaves the peptides or peptide chain from the amino end, and carboxypeptidases, they cleaves the chain from the carboxyl end. So here the carboxypeptidase cleaves the peptides or peptones from their carboxylic end side, from here end side, and aminopeptidases, they cleaves the long chain into smallest peptides. So whatever the peptides are there, those peptides are breaks into tripeptides by these aminopeptidase and carboxypeptidase. So here tripeptides and small peptides, small chain peptides are created. Small chain peptides are created by the action of aminopeptidase and carboxypeptidase, and these tripeptides are converted into dipeptides by tripeptidase, and the dipeptides are converted into amino acids by dipeptidase enzymes. So these are the enzymes and action of the protein digesting enzymes on the proteins present in the small intestine. So in the small intestine, proteins also completely digested. So first we discussed the digestion of carbohydrates. They released the end products are glucose, fructose and galactose. And secondly, the protein metabolism, protein digestion here. In the protein digestion, say here amino acids are created. So the end products of the digestion of proteins in the small intestine ileum, those are the amino acids. Up to now, any doubts? If you have any doubts, please ask me here. So okay, we stop here. Tomorrow we continue our topic. So every day we have live session from 9 p.m. to 10 p.m. and we have our own app is present, that is Kumar Institute of Zoology. If you have any doubts, call on this number 83310999266, this number. Okay, if you have any doubts and take clarification from this number, and if you have any doubts regarding the zoology and this topic, please chat me. Okay, and now we are closing our session. Okay, if you have any doubts, please ask me. Okay, we are closing this session, and tomorrow we continue the digestion of fats, the digestion of nucleic acids, RNA, DNA. So all we discuss tomorrow session. Okay, and don't forget to share this link, Zoology Online Guru. Okay, like, share, and subscribe my channel, Zoology Online Guru. And also we have another Telegram channel. Also we have the Telegram channel name is also called Kumar Institute of Zoology. So like, share, and subscribe my channel. Thank you. Good night. Okay.